if we grow the mutant library we got in any particular stress we want, and without stress, then we can compare what genes is required in what particular condition. So in this cartoon, you can see on the right, gene X has become essential because of the stress condition, and it was non-essential with no stress. But gene Y, I want to bring your attention to gene Y, where there are no insertions already. So how can we find out if that particular gene is required for the stress condition or not? So here comes the expression part of TRADS. So we introduce the outward facing promoter in each transposon. So when they are inserted next to the essential genes, and if the essential gene is required in that particular stress condition, it will be overexpressed. And if it's not required or it's detrimental, then it will be repressed. So in TRADS, when we don't have outward facing promoter, we can just only inactivate the genes. And in that way, we also we can measure the different genes in different stress conditions, but we will not be able to measure the role of essential genes. But in TRADS Express, we are measuring the inactivation of the genes, but we also measuring the overexpression and repression of the genes. So here is some of the data I have shown with the 4 conditions like A, B, C, D. A is the protection, where particular genes become essential when the stress in particular stress condition, and B is where a gene was essential or semi-essential in no stress, but its knockout is better for the survival in that particular condition. And overexpression, when there is essential gene, but that particular gene is required in stress condition, it will be overexpressed, as shown in the C panel. And repression, when particular gene's presence is detrimental, but is essential gene, so it cannot be knocked out like B. So it will be repressed by the downward insertions. So there are some more data slides. So each line is showing a particular TRADS experiment, and the top 2 are controls, and the bottom are the conditions with different concentrations of the same chemical or same stress. And the experiment was done in duplicates. So we can see that this particular gene has become essential in this stress condition. And this is example of TRADS Express, basically, where the expression of the essential gene is required to survival in the stress condition. So when we have dozens of these graphs and we have loads of data, so it's hard to make sense of the data by just looking at it visually. So our bioinformatics team has written a software, AlbaTRADS, that helps us to understand the data better. So it produces a gene list that are important, and it also points out in the form of like heat map or network, so that we can follow the particular metabolic pathways. So what are its applications? So when we have the new antibacterial compounds, or like I'm pretty sure like in Pakistan, many students during their master's or PhD, they do like extract antibacterial susceptibility tests and honey or garlic, etc. But they stop after doing MICs, and they don't know what is the mechanism of action of that particular compound is. Or they have synthetic compounds, and they have found that they are susceptible. These compounds are susceptible, showing susceptibility against certain bacteria, but they don't know the metabolic pathways or the mechanism of action of those compounds. So where the TRADS Express comes in, and it can assay the whole genome and can tell that basically what genes are important in what particular condition. And this is one application. We can find out in other stress responses like high and low pH, high and low temperatures, biofilm formation, competition assays, and there are multiple applications of TRADS Express. So today we will be discussing one of the chemicals, known one. It's triclosan, and Triclosan is an antibacterial agent, and it is used in loads of cosmetics and other household products. So the purpose of assaying triclosan in the through TORUS Express was to find out if it identifies all the possible resistance or susceptibility mechanisms of triclosan, then of course it can identify the susceptibility mechanisms and mechanisms of action of the new compounds. So now what we did is in this particular condition, we know that already from the literature that in oil acyl carrier protein reductase, that is basically lipid formation, Fab-I, its involved in lipid formation is essential for that triclosan is a target for the triclosan. And there are some other resistance mechanisms, but they are like showing minor play. So what we did is basically we competed multiple mutants. So I always give this analogy, like it's like a marathon. We start with hundreds of thousands of mutants or the starters, and then we try to find out who is the winner, who is the gene or the set of gene that are essential for that particular stress condition, who are the losers and who are the winners. So in this experiment, we did 8 different conditions of triclosan, and we did control, of course, and solvent control as well. And that makes up to 66 experiments. So this is a reproducibility of TORUS. As we can see that it's pretty reproducible with the correlation value of 0.97. And this is the distribution of insertions shown on the whole genome. And it is the insertions are almost uniform all around the genome. So here is the Fab-I on the right-hand panel. We can see that overexpression of Fab-I has happened here in this triclosan stress condition. And this is, we know from the literature already that it should happen. So it means our TORUS Express is working. And another pump gene, ACRB, has become essential. We already know about this from the literature. So this gene, if it is knocked out, the cells perform worse. So they will not survive. That's why they have shown that there are insertions in the no stress, but in the stress condition, there are less insertions, showing that this gene is essential in the stress condition. And we have find out by doing the growth assays in the Q library, where we have the knockout of ACRB, and we have compared it with BW, that's the wild type. And we can see that at this particular condition, ACRB mutant cannot grow, but BW wild type is growing. So this graph shows that our validates our TORUS data. Here, another example of that. So YHBJ gene is knocked out in this particular stress condition of triclosan. And as we have the knockout of YHBJ gene in our Q library, we compared it with the BW, that's wild type. And we can see that knockout performs better as compared to the wild type. And we find out some new stuff. So for the transporters, and there are like TRK, A and H transporter system is involved in the transportation of triclosan into that. This is the new stuff that we find out through triclosan that is not available, that wasn't studied already in the literature. So we can see that like there are multiple genes we identified. We identified almost 70 genes, and that involved in almost 7 different pathways. And we can see that it has a sidal and static mechanism by looking at the network. These low concentrations, they are clustering together, and the high concentration, they are clustering together. And this can be shown on the heat map as well. So this cartoon shows, summarize basically whatever I've told you. And I won't explain the whole cartoon, but I will bring your attention to this TRK HNTRKA here. I hope you can see my cursor. So this is involved in the transportation of triclosan in the inner membrane. And on the left-hand side, bottom left-hand side of this cartoon, we have shown Fabi how it is involved in the synthesis of phospholipids. So in short, we have we've shown that TRADOS Express is a powerful genomic tool, and TRADOS Express now allows the whole genome to be assayed. That was not the case in the past. And the refinement at QIB allow experiments to be done at a scale and analysis across genomes. And we have a significant interest in applying this in diverse projects and with multiple partners. And we are entering into the microbiome health space. I would like to acknowledge my team here at QIB and our collaborators. Thank you very much and for listening. And if you have any questions, you're welcome. And if you are interested in this TRADOS Express, you can contact Fayaz, and we're happy to help on that.
That's great, Yasir. Thank you so much. Is there any question from the audience? House is open for the questions. I think there is no question from the audience. Yasir, I have a question that can you just a little bit explain, because TRADOS, that is a very, I think, a difficult thing for the chemistry people especially. Can you just give us a few applications of the TRADOS in the food chain, and a few applications which are related, might be related to chemistry?
So might relate to chemistry. So we are trying to understand microbiome, that is basically the community of bacteria. And we know that with the consuming chemicals like triclosan, that is present in a very small concentration in the households, and multiple products, you can see that cleaning products, and etc. So that can affect the microbiome of our gut and skin and our oral cavity. And why it is important, why if it affects that, why should we bother about that? Because as we have seen in the gene network, that triclosan resistance mechanisms are overlapping with antibiotic resistance mechanisms. So we found out that if a person is carrying normal flora or microbiome that is resistant to triclosan, can become resistant to particular antibiotics as well. This is one of the aspects. The second aspect is when we study it on the biofilm formations, and as we know that in food chain, we have many bacteria that can make biofilms on many kind of frozen foods or packed foods, or like fresh, even available foods that are sold in the market, very thin biofilm. And we were trying to understand how these bacteria make biofilms. And if we know how they form the biofilms, then we can stop it by finding the appropriate chemicals and or chemical inhibitors. So if we know the set of the genes required for that, and then we can predict it through computer algorithms, what kind of chemical structures would be needed. And there the chemistry people come in, and they provide us with particular set of libraries that are inhibitory to those pathways. I hope I have answered your question.
Yeah, thank you so much. And can we use other certain chemicals or the antibiotic, and then we can apply this TRADOS technique? Especially chemists, they are involved into synthesizing a lot of different compounds. And then either they can check the effect of those compounds like the triclosan on the different bacteria and check their resistance or anything else in the genome.
Exactly. So I think this is a very good question, Faz. I think my aim was, when I was talking about triclosan, I haven't selected any other stress conditions at the moment, because I was thinking that I'm talking to a chemistry department, so they would be generating loads of chemicals. And as I know from my experience that there are many different libraries are generated during different experiments in the chemistry, and when they are looking for antibacterial compounds. And what happens, most of the experiments stop at the susceptibility level when they check that this particular compound is showing some action against, let's say, staphylococcus or any gram negative like E. coli or Pseudomonas, and there is a blockade. We cannot go further down in the stream because uh, we don't know where to start, and we cannot assay every gene and every possible aspect with like individual experiments. That would be hugely expensive and labor intensive. But this TRADS gives us the opportunity to assay the whole genome of any kind of chemical we have generated, we are interested in, that has shown susceptibility, and we want to understand like how it works. Does it has the antibacterial action similar to the antibiotics in use, or it has a unique antibacterial mechanism? So we can find it out very easily through the stress conditions, as I have shown here. So we can grow it, the library, with the stress and without stress. So when we grow them, then we can compare the genes that are required in the new chemical, and then we can find out, we can compare the data available for the antibiotics. So we have done the experiments with multiple antibiotics, and some of our papers are already out there, and others are in print. So we can see that we have identified the susceptibility patterns or the pathways that were about 10 to 20 years worth of work in just one experiment. And I think that is very cost economical, and it's very robust. It's not very labor intensive, and of course we can validate it through the knockouts or the real other individual experiments we want to do.
Thank you so much, Yasir. That's very informative, actually. From audience, there is a question that what is the meaning of stress here?
Oh, okay. Sorry, I should have explained. So when we say stress, it means any kind of pressure. So antibiotic is a pressure on the bacteria. And if we say that temperature, like if we change the temperature like from 37 to 55 degrees or 60 degrees, it is a stress condition, because we are imposing not the normal condition or the most favorable condition for the bacteria. We are trying to find out the survival in the extreme conditions. So I think anything where we are trying to add a chemical or a barrier on the bacterial growth, that can be said as a stress, and it could be very minute and it could be huge. Like we can say that 39 degrees is a small stress of in the temperature as well, in the sense of temperature. If we're growing the bacterial, normal growth temperature is 37, or the optimal growth temperature is 37. And if we're growing it on 39, then we will say that we are trying to find out how the bacteria behave in this temperature stress. So I hope I have explained. So basically, it could be any antibiotic, it could be any chemical, it could be variation in the optimal growth condition. So that is called stress. So I should have explained in the
Thank you so much. Thank you so much.
You're welcome. Thank you very much for listening.
during this session, and I will request Professor Dr. Farooq Anwar.
There is
Sorry,
a
can you
message, I think, sir, we can use it for pesticide and fungicides I think in the chat.
Yeah. There is another question, Yasir, from you. Can we use it for the pesticide or the fungicide?
Sorry, Fayaz, I interrupted you while I was reading the message. Well, you were saying something.
It's fine. Just proceed.
Okay. Yes, of course. We can go for the pesticides and fungicides. So there are limitations in that sense. When we are talking about, fungicides, we need to have a mutant library in that particular organism we are interested in. So if we have a library in any kind of fungal pathogen available, then yes, of course, we have some colleagues in Glasgow who are working on this. So they have made a library and they have assayed it. So yes, it is equally applicable on the fungicides. In case of pesticides, as we try to find out the pathogens, if they what kind of target pathogens for that particular pesticide. If it is a fungus or a bacteria, yes, we can use Stardust Express. If it's a virus, it will be a bit difficult to say at this point, because we haven't studied viruses using this method. And if there's the plants, if we want to study the plants and how they behave, because when we add pesticides to the plants, they are under stress as well, because it's not normal for the plants in the organic conditions to have pesticides on them. So if we want to see the effects of pesticides on plants, then we need to have a library of mutants of the plants. So some of our colleagues are working on that. But yeah, your question is quite valid, and the answer is yes, we can do that. How this is useful for organic chemists? As I have mentioned, like organic chemists, they are very essential role in the development of new antibiotics, because they are the people who tell us how we can change or rotate or particular functional groups or do the things like that. And whatever we find through our I won't say the targets, but the metabolic pathways that are important for that particular stress condition. If we want to interrupt that metabolic pathway, and we know that this is the target, basically, if we could interrupt this metabolic pathway, this will stop the particular growth condition or the growth of the particular pathogen. Then we contact chemists and we try with the available chemical and start with the available chemical, and then we will request them, is it possible to make variations of this, like adding methyl or other functional groups onto that. And if it is possible, then we go for that. So I think organic chemists, they have a very essential role, and it is a kind of very complementary method for them if they're interested in pursuing this road or pathway. I hope I have answered your question.
Thank you, Yasir. Thank you so much. That's really very helpful. And I think let's move towards the concluding session. I will request Dr. Muhammad Iqbal Bhangar or Dr. Nizamit, if they are still here, can you just give some comments on the symposium? Yes, sir. Dr. Iqbal Bhangar or Dr. Nizamit Sari, can you give some help? Dr. Bhangar, thank you so much.
Professor Dr. Farooq Anwar, Professor Dr. Fayaz, and all my related colleagues who have spoken in this seminar, plus the most valuable are the participants, and especially our students, a young age, who have entered into the research. This organization of this seminar is so fruitful, especially as I said in the beginning, in these days of pandemics, that we don't have a chance to face-to-face meeting. So we cannot stop the research process and say that everything is stopped, so we have to. So this is a blessing that these software is available, and we can hold seminars, international seminars, much more easier than holding them otherwise, because you can call speakers from America, you've seen speakers from USA, speakers from UK and Malaysia and Pakistan, all participated and without any big problem. So, but definitely the organizers have a role to play into this one. And the other thing is the students that I have seen the keen interest of our students who have participated into this one-day seminar, symposium, the way in which they have interacted with the speakers. This shows that they are very much interested in their research, and this provides them an opportunity to actually enter into a sort of a linkage or a collaboration with these people who are working in USA or who are working in UK and Malaysia or in Pakistan, so that the facilities that you have in uh, Sargodha or Faisalabad, you can also get the facilities from abroad, or certainly collaboration, research collaboration is of prime importance, especially for our students. And I'm sure that the efforts made by your dynamic director, Professor Farooq Anwar, and also Dr. Fayaz will go a long way, and we'll see many more such seminars and conferences on multiple subjects being held more frequently, because as I said, it is easier to organize this rather than to organize a face-to-face seminar in which you need a lot of funding and a lot of arrangements and a lot of approvals, if not, and so it is very good that if you can organize this, say, every 3 months or quarterly or every biannually, so that our students will get an opportunity to interact with their web. I wish all of you a great success. Thank you very much.
Thank you, sir. Thank you. Thank you, sir. Thank you very much. Is Dr. Professor Zaman, if he is available, we would like to have his valuable comments. Professor Zaman, I think he left. Sir, we can have the honor. Indeed, we have a great opportunity to have you as a speaker, and all the other speakers, they also delivered very valuable talks, which were very fruitful, and especially Professor Dr. M.I. Bangar, my supervisor. I feel great honor to be his student, and I learned a lot during my PhD studies, and he treated us just like children. So he imparted us many values of life. So I am always indebted and always thankful where I am. I am all due to his kind supervision and his kind support. Thank you, sir. Thank you very much. I am also greatly thankful to Professor Zhu, who have presented his lecture in the morning although it was off time in USA. He is our great Chinese friend. I am also thankful to Professor Nazamad. I spent 1 year with him in Malaysia during my postdoc studies. He is a great researcher. So I am greatly thankful to Professor Nazamad. And two speakers we were lucky to have from UK, Dr. Yasser and another speaker, Dr. Sharma. So I am also thankful to the speakers from UK. I am also thankful to all the participants for their very active interaction and very active participation, which makes this symposium a successful event. I am also greatly thankful to Dr. Fayaz, and I much appreciate his great efforts in arranging and organizing this sort of activities. He is a very active researcher and working at Institute of Chemistry. And I am greatly thankful to him and his team. At the last but not the least, I would like to again extend my very sincere thanks to all the invited speakers, honorable my teachers, and all the participants who have make this event a success. Thank you very much, and Allah Hafiz. God bless you. Thank you, sir.
Thank you so much, sir. Thanks to all.